Today is Thursday, September 23rd, 2021. Before we get into the new material, let's talk some old material. I literally, 40 minutes ago, finished grading and curving your lab and lecture exams, so snuck them in just under the wire. Thankfully, I was able to get that done so I can drink heavily tonight. And the results are, well, let's say interesting. The lab exam, the average was a 50.69 out of 82, which works out to about a 62%. The lecture average was a 68.7 out of 100, which of course is a 68.7, rounds up to 69%. But as anyone who's ever taken statistics or maths know, there's a lot more to numbers than just averages, and this is a great example. Notice here I've put the high scores and the low scores up here, not so that two people feel really good about themselves and two people feel really bad about themselves, but to really emphasize that even in a second semester class like this, people come into this class with different expectations. Different people had different 430 constructors, different 430 experiences. For some people, 430 was just this past summer where you were rushing through the material. For some of you, it was months ago when you took it in the spring or even possibly last fall. Because of that, there tend to be very diverse expectations of what the exams in this class are going to be like. For some of you, clearly these exams were very different from what you experienced in the first one, and these wide range of scores show this. Now, there are two important pieces of information here also on the board. The first is that thankfully grades in this class are not determined solely on your exams. It is all the points. All the points in this class, every point counts the same. 1 point on a lab exam counts the same as 1 point on a homework, counts the same as 1 point on a Physio X, and so on and so forth. So the good news is with all the grades thrown together, all the points thrown together, the average is pretty good. A 78% is not a bad place to be after the first exam. However, the other important thing to remember is that we are going to finish this class with probably somewhere around 1,200 to 1,300 points. So notice we're only 350 deep. So the two people who did really well in this class, can they just sit back, rest on their laurels, take a break, vacation between now and the end of the class and just slide into the A that they want?
No.
No, of course not. And by the same token, the people who are at the lower end of this spectrum shouldn't grab their whip and go to the bathroom and flog themselves either as a result of this. Use this first exam as a learning experience. The good news is there's no more surprises. The same way I wrote this lab and lecture exam is going to be the same way I'm going to write all the lab and lecture exams. The same expectations I had on this exam are the same expectations I have for the rest of the exams, right? And as I've said many times, and I'm sure you're tired of hearing me say, people often lose points, not because they don't read the information. I mean, they don't know the information, but because they don't read the questions carefully. There are plenty of people, for instance, on the lab exam who lost partial credit because they forgot to put right and left, even though I continued to tell you that. Or they forgot to put artery and vein, even though I continued to tell you that. Or they identified the blood vessel instead of identifying the blood vessel that receives blood from it. Make sure you read the questions so that you can answer them correctly. Make sure you scan your area thoroughly to show me all the stuff. Make sure your area is clear of materials. There should not be whiteboards or post-it notes or any other type of materials around your environment when you are taking this. So there were some notes put into some of your exams for those things as well. All right, so again, the good news is regardless of how you did on these first exams, every single person in this class is still capable of getting an A. Right? If you did well, then be confident in the fact that you attacked this material the correct way. And if you continue to attack the material in this way, you should continue to be successful. If you weren't as successful as you wanted to be on this exam, then appreciate that you didn't attack the material in the correct way. But the good news is there's still time to change. One of the things I want to do to help you to change and to help you to improve is while I released the scores about 40 minutes ago, I have not released the exams yet. I will release the exams after class. If I released them before class, then nobody would pay any attention to my lecture. You'd be all looking at your scores in your exams. So I'm not going to do that. They will be available for about 24 hours. So for the rest of today and till tomorrow afternoon, they will be available for you to be able to review them. If you're not able to review them in that time, or you have further questions, or you want to see them again, you're welcome to come to my office hours. I will happily go over the exam with you to help you to see why you lost points or why you got points, what types of things I was looking for, and how you can improve it. And do it now, while there's still close to 1,000 points left. Occasionally I have a student who comes up to me right before the final exam and says, what can I do to improve my grade? And unfortunately, at that point, the answer is really not much, because we're already 1,100 points into 1,200 points in this class. Now's the time where small changes can make huge differences in your grades. Now is the time to adjust how you're preparing so that you can be successful moving forward. With changes, every single person in this class is capable of getting the grade that they want. So do the work, do the due diligence, learn from this first exam to help you to be successful. All right, questions on that?
Professor, the high scores for both the lab and the lecture exam, were those the raw scores and then they
No,
were?
great question. Great question. No, these are the curved scores. So yes, these scores are already curved. So both of these scores have been curved. All right. Great question. Any others? All right, then let's talk about our game plan. Today, we are going to finish our discussion of the immune response, our body's defenses. And then at the end, we'll talk a little bit about what the expectations are going to be for you on the endocrine system. You have 5 whole days before we meet again. And while a lot of that time should be used, understanding and mastering the material that we've covered in the lymphatic system and our body defenses. But it will also behoove you to look forward towards the endocrine. The endocrine is going to have a massive amount of histology. There's a lot of chemical and cellular interactions on these that make it some of the more challenging material because it's least intuitive. You can't hold it in the hand the same way you can hold a heart in your hand. So I would encourage you to get started on that, and I'll give you some tips to help you with that as we move forward. Again, only 2 assignments left in this section. Again, you should be working on your group presentations, so I haven't given you as many assignments, but you do have 2 lab simulators. Your PhysioX exercise 12, only the 3rd activity, and the Labster Introduction to Immunology, which, remember, for full credit, you need to complete 100% and get at least 80% correct. Remember, I guess technically you have 1 more homework assignment associated with your group presentations, and those are the outlines. Again, as soon as your outline is done and you are happy with it, I encourage you to submit it. Remember to make sure you have everybody's name on it, make sure you have the references on it, and of course the name of your topic should be on there as well. But you have until Friday. Again, I don't like having assignments due on days when we're not in class because it's easy to forget. But there is no leniency on this. If it is turned in at 6:05, it is late and you are being penalized for that. So make sure you get it done, make sure you get it on time, put it in your phone, or better yet, as soon as you get it done, submit it well before Friday. Pretend it's due Thursday before class, and then you can get it done and submit it and not have to worry about it. But for those of you who need the extra time, I'm giving you the extra time. Just make sure it is turned in on time. because I want them available over the weekend for people to review. Because on Tuesday, we have group presentations, and then 2 days later, we have the lab and lecture exams. So the same way I post my lecture slides ahead of time so that you can prepare for the lectures before we have a chance to talk to them, I want to post the outlines for the presentations so that those people who want to prepare ahead of time can. So they don't have to try to cram it all in into the 2 days after the group presentations and before the actual exams. All right. Questions on any of that? Excellent. Stunned silences. Love it. All righty. Let's move on to our lecture then. We left off last time and we were talking about our third line of defense, which is basically the same thing as saying our specific or adaptive immunity. Again, it's specific based on the antigens. It has that versatility, effective, systemic, has the memory. But remember, we talked about the downside of it is it takes time. It is a slower process. As we kind of already discussed when we were talking about the blood types, the way we're able to precisely identify harmful pathogens is by the tags that are on the surface of all of our cells. And those tags, like we learned when we talked about our blood types, are called antigens. They're mostly proteins, but they can include carbohydrates or lipids or some combination of those things. And when we talk about these antigens, we talk about antigens as being complete or incomplete. A complete antigen basically has two main requirements to it. The first is what we call immunogenicity. Immunogenicity is basically the ability to stimulate a specific lymphocyte to divide. As we know, we have lymphocytes. We've mentioned that they're part of our immune response. We haven't talked about their function yet, but we will. And they basically have antibodies on their surface that have a particular binding site on them. So if the right shaped antigen comes in and binds to it, so here's a pathogen that has that triangle shaped antigen on it, that is going to stimulate this lymphocyte to divide, to produce massive numbers. Oh, let's make them purple so they're the same. Massive numbers of new cells. And all of these new cells that are produced are going to have that same specificity to them. They're going to be able to also be associated with that same triangle shape antigen. All right, does that make sense? Excellent. So the second half of being a complete antigen, and I'm gonna have to erase this for a second, but I will do this before we erase it. Let's think about this. This antigen, whoops, this pathogen has all of these triangle-shaped antigens on its surface. So all of these new cells that have been produced, should they be able to respond and attack that antigen in some way? This is the part where you all say yes enthusiastically.
Yes.
Excellent. I'm not sure if that was enthusiastic, but we'll take it. There was some enthusiasm in it. Absolutely. And that is what we call reactivity. Reactivity means that it is able to react to those activated lymphocytes and those activated antibodies. If an antigen has both of those components to it, then we call it a complete antigen. If it doesn't do both of these things, then we call it an incomplete antigen, or what is also known as a hapten. The problem with a hapten is that it can be reactive. So what can happen is there can be a very benign molecule. That very benign molecule might be, for instance, the pollen that is released from cut grass. That by itself isn't able, if we go back and grow that lymphocyte that we were talking before, here we have a lymphocyte, and that lymphocyte has that triangle shaped binding site. Is this going to be able to bind to that triangle shaped lymphocyte and stimulate its reaction? I mean, stimulate it to divide?
No.
No. The problem is these antigens will sometimes bind to other cells or other proteins in our body. So let's say I've got this cell here and just happens to have this funny shape structure on its surface. And when this hapten binds to it, suddenly these two things together take on a triangle shape. Now, again, obviously these things aren't really triangle shapes, so I'm oversimplifying this a little bit. But notice the key that what happens is that when that incomplete antigen or that hapten binds to a protein or a cell in our body, it becomes reactive. And so any of these lymphocytes that might be here have the ability to attack it, have the ability to cause an inappropriate response. We call this type of inappropriate response a hypersensitivity. Or what's the other term we give to this type of a response?
Allergies.
Exactly. An allergy. Exactly. That is exactly what's happening here. What happens is that hapten binds to some protein or some form of cell in our body. And suddenly, as a result of that, it becomes reactive. And now it can trigger that inappropriate immune response. Like I said, it's frustrating when it's that pollen from that cut grass. And so as a result of it, you stimulate the release of histamines in your mucous membranes, increasing mucus production, increasing blood flow, which causes it to swell, causes mucus to run out of your nose. You get all congested, you get a runny nose. You know, your eyes get watery, gets to be a pain in the butt that way. It's a little bit more serious when it's, let's say, oh, I don't know, a piece of protein from a peanut that suddenly causes a massive vasodilation of the blood vessels, causing a drop in blood pressure, or causes a massive increase in mucus production and swelling of the mucous membrane in the airways, meaning that that person can't get oxygen and they can't to their lungs, they can't get blood to their brain. And someone remind me again what we call that process?
Is it anaphylactic shock?
Yeah, anaphylactic shock, absolutely. Anaphylaxis, anaphylactic shock, absolutely. So these hypersensitivities, unfortunately, have the potential to be incredibly dangerous.
Professor?
Yes.
So just to clarify, the pollen itself has antigens that bind to our proteins?
No, the pollen itself is an antigen.
Oh, I see.
It's a protein, and it's a protein that normally is benign, right? That's the key to this. These are small molecules that are floating around in the air around us that normally have no effect at all. But for some people, when it binds to another chemical or another cell in your body, it triggers a reaction.
Got it.
So
Okay.
you could have 100 people in a room with all these little molecules floating around in the air, and maybe 2 or 3 of them, it binds to just the right, or I guess just the wrong protein, depending on how you think of it, and they get that reaction and they have that allergy as a result of it.
Got it. And because they are reactive to that, that's all based off of their genetics, right?
That is a great question. Again, this is something that I'm personally very aware of because my daughter has a severe peanut and tree nut allergy, and she's the only one in our family who has it. So clearly that seems to be an indication that it isn't genetically related. The causes of these things are not fully understood. As many of you, and in fact, I'm going into this now because sometimes one of the groups picks hypersensitivities as allergies as their topics, and nobody did. So that gives me the freedom to talk about it a little bit more. But as many of you are learning about on your own disorders and diseases that you're learning about, the cause for many of them isn't understood. You know, some of them may be related to getting a particular viral infection at some particular stage in their life that may have caused it. You know, what causes, autoimmune disorders where the body attacks themselves. We don't fully understand. Many of these may have a genetic component, but others may not. And it may not be a direct genetic component. It may be a genetic sensitivity, right? Where if a person may be more sensitive to it, so that if they're exposed to the right environmental cues, that could cause it to occur, right? That was one of the thinking at the time when we found out about my daughter's allergy. That at that time, it was believed that being exposed to a protein, to a peanut protein in the first 2 years of life increased the likelihood of a peanut allergy. So nobody was supposed to give peanuts to their kids for the first 2 years. Now they've reversed that, saying, no, you need to give it to them in the first 2 years. Much of the research is counterindicative, and a lot of it's not clear. So many of these causes we don't fully appreciate and understand.
Okay, that makes sense. Thank you.
Now, some allergies like hay fever and things like that, there may be a stronger genetic component to them because they're less severe. I don't think they're studied as closely. But again, since most families that have hay fever tend to live in the same areas, again, is it because they're living in Sacramento, or is it because they're all genetically related? So again, I'm not sure. I'm not sure if I don't know the answers, and I'm not sure if anybody else does either.
Thank you.
Yep.
Professor?
Yes.
I had a similar question for the autoimmune disorder, but when I was doing the research for my presentation, what I understood is there are genes, but they can be more common, but the main problem are not the genes, but when they are actually being expressed. So the environmental factors are the main cause for those.
Yeah, and like I said, that more and more it is appearing that those are the types of things that, again, you know, if you think back to the 50s and 60s, it was that whole big debate. We talked about this last class. Is it nature or is it nurture? Is it genes or is it your environment? And we're finding more and more that all of these things, both good and bad, are a combination of both. So like you said, you may have a gene that makes you predisposed or more likely to have X happen to you, but X is only going to happen if, you know, these environmental events occur. So yeah, absolutely. So and so again, it's not surprising is if as you are going and doing your research that they don't have a boom, this is the cause, right? For some things they do. For some things they know this gene, this mutation, or this bug with this chemical, or this virus, right? Some things we know the causes, but many of these autoimmune disorders and immunodeficiencies, we don't fully appreciate and understand the cause. All righty. So let's move from this back into our third line of defense. Again, we kind of understand about antigens and antibodies, so we'll talk about those some more. And as we finished off the last class, we talked about all the different types of immunity. But the one that we typically think of as our immune response, what was that specific type of immunity again?
Innate.
Was it innate?
Second line of defense.
Active.
Now, remember, we talked about, so innate was getting closer. We talked about innate and acquired. And then in the acquired, we had different types.
Active and passive, right?
Sure, active and passive. And even under active and passive, we had different types.
The naturally occurring.
Naturally occurring and induced. So put all the pieces together. Of all of those combinations, what was the one that we said was typically what we mean when we say our immune response?
Acquired active immunity.
There we go, exactly. It is that active, naturally acquired immunity. Exactly. Excellent. Now, when we talk about that, and again, remember, that is the one where we are exposed to the pathogen naturally in our environment. That cold virus goes up my nose. And then as a result of that, I get the symptoms, but I do the work and I get the memory cells. Right? That is what we think of with the immune response. Now, there are two different components to our immune response. The first is what is known as the humoral immunity, because it only happens in the humerus, right? What does humoral refer to?
Liquids of our body.
Yeah, exactly. The liquids of our body. Primarily, that means blood and interstitial fluid, but also lymph and things along those lines as well. Humoral immunity is also known as antibody-mediated immunity, because that's the part that uses antibodies. That's convenient. The second component of our immune response is cellular immunity, or cellular-mediated immunity. With antibody-mediated, basically what we have is we are producing antibodies for pathogens directly. Right? That is what we're doing with our humoral immunity. Whereas cellular immunity is actually cells of the body attacking foreign or abnormal cells. So basically it's cells attacking other cells. So we have these two different types of immunity that are giving us two different types of defenses. Now, we are going to talk about the cell-mediated immunity first, because it is the one that typically happens first. But before we do that, we need to talk a little bit about the cells involved. We've already know our lymphocytes are the primary cells involved in our immune response. We learned that back in the blood system when we were talking about the cardiovascular system and the blood. Someone remind me again where lymphocytes originate?
Monocytoblasts.
Okay, so that's one way. Okay, that actually wasn't the answer I was looking for, but you're absolutely right. So they start from a pluripotent stem cell. That is the hemocytoblast.
Oh, sorry.
That's all right.
Is it the bone?
We can go this right to it. Yeah, closer. But let's finish this first. That then divides into a secondary stem cell. What is the secondary stem cell?
Lymphoid.
Lymphoid. Excellent. But you're right. This occurs in the bone. Although, can we be more specific?
The marrow of the bone.
Yeah, bone marrow. Excellent. Excellent. I see you wrote that too, Amy. Awesome. Excellent. So there you go. It occurs in the bone marrow. Spectacular. So there in the bone marrow, we have those pluripotent stem cells, the hemocytoblasts, divide and become lymphoid. Oh, I forgot the P there. Lymphoid stem cells, which then become lymphocytes. And they become immature lymphocytes. Again, by immature, we mean that they are not capable of an immune response. you can take a virus and rub it all over it, up and down and all around, and it will do nothing in response to that. It is not capable of responding to a pathogen. So it needs to go someplace to mature. And it turns out there are two places it can go to mature. It can actually stay in the bone marrow and mature. And the ones that mature in the bone marrow become B cells. And what did we say the other place they could go to mature was?
To the thymus.
Thymus, absolutely. And the ones that mature in the thymus, what do they become?
T cells.
Cells.
Exactly. Thymus. And those become T cells. The B cells, the one that develop in the bone marrow, are the ones that produce antibodies. So they are responsible for our humoral response. Whereas the T cells are the cells that directly affect and attack other cells. So these are the ones that are responsible for the cell-mediated response. And now that I've written all of that, I have to erase it because we got a pretty picture that goes along with this. Oops, oh no, I have that in a second. Okay, we'll talk about that in a second. So let's not erase that, let's leave that. Once they mature, we say they are immunocompetent. Remember, as we talked about, immature means that they're not capable of immune response. They cannot respond to a pathogen. Mature means that they can respond to a pathogen. However, every mature lymphocyte we have in our body, has it necessarily come in contact with the pathogen associated with it? You remember I mentioned in the last class, many of you probably have lymphocytes in your body that could protect you from malaria. However, has every single person who's had that actually been to Panama, been through the Panama Canal and gotten malaria while they were there?
No.
No. You could have that cell in your body that's going to protect you from malaria, and you may never come in contact with malaria, which means that that cell will never get used. So mature means that it can respond to a pathogen. But if it has not responded to a pathogen, then we call it naive. So a naive cell is a mature cell that has never been activated. And what that means is that it has never come in contact with its specific pathogen. And let's take this one step further. How do we determine what pathogens our cells are prepared for? What determines whether or not you have a cell in your body right now that is there naive, but if you should ever come into it, will protect you from malaria?
Antigens?
No. Why do some people have cells in them from birth that can protect them from malaria and
Genetics.
others don't?
Genetics, like came from
Genetics, absolutely. Remember, your starting point, which cells you start with, are determined genetically, right? That's that innate defense we talked about. I'll keep beating that horse. Our starting point for all of this, which cells we have, what shape antibodies we can produce. All of that starts by what we have in our genes. Our genes determine our starting point, which ones we have and which ones we don't. All right. Questions on that?
Does that mean that like somewhere down the line in our family, someone was exposed to malaria, for example, or it can just be in your body?
Both. Right? Think of the Indians, the Native Americans, sorry. With the Native Americans, when the colonials came over, right? They were super happy to see the Indians, so much so that they gave them all these wonderful blankets. And what were so special about the wonderful blankets they gave the indigenous people?
They were diseased.
Yeah, they were filled with smallpox, a disease that most of the indigenous people had never been exposed to before. And so most of them died. But did all of them die? No. Some of them had a genetic predisposition where they had some cells that were able to protect them from smallpox. That doesn't mean that, you know, many years ago they were exposed to smallpox. It just means that they had the right genetic mutations in their DNA that allowed them to be more resistant to that. So yes, it can mean that someone down the line had it, but it also could just be a genetic variation as well. All right. Isn't history wonderful? All right, excellent. So let's talk a little bit about how we make these cells immunocompetent. This is most closely studied in our T cells, but as far as we understand, it works the exact same way in B cells as well. This maturation of the lymphocytes takes place over 2 to 3 days, and it is a 2-step process. And let's go to the whiteboard for this, because I think this is something that we can simplify and look at visually and make some sense of. So the maturation of the T cell. All right, so we need our starting point. We need a T cell. And we basically need a cell of our thymus. And let's not even call it a T cell yet. It's an immature lymphocyte. All right. Now, what we want our T cells to be able to do, remember, is to be able to recognize stuff that is us. To do this, it must be able to, well, let's say it this way. How do I want to do this? It's here. This here. Oh, I like it. Let's do it this way. Hold on. Down for a second. All right, to be a successful T cell, A T cell has to be able to do two things. The first thing it needs to be able to do is bind to a cell. The second thing it needs to be able to do is recognize the antigen. So to be a successful T cell, these are the two things that have to occur. You must be able to bind to a cell, and then you must be able to recognize the foreign or abnormal antigen. All right, we comfortable with that? That makes sense? All right, let's talk about how this works. The first step, step one, is what we call the positive selection. To be a successful T cell, the T cell must be able to bind to another cell. It does this with a special protein that is called the major histocompatibility complex, which we'll now abbreviate MHC. So our cell on our thymus is going to have a special protein on its surface. And that special protein on its surface is the major histocompatibility complex. So what our T cell needs to be able to do is it needs to have the correct binding site to be able to attach to that. So it must have a binding site that is going to allow it to bind to that major histocompatibility complex. And if that occurs, then it passes. However, let's draw a second lymphocyte. This second lymphocyte with this second protein, would it be able to bind to that major histocompatibility complex?
Not completely.
Not
at all?
completely, exactly. And so if it's not able to completely do it, then it is going to fail. Because this one will not be able to stimulate a response. And if a white blood cell can't stimulate a response, is it going to be of any use to us?
No.
So what do you think we need to do to this cell?
Destroy it.
Kill it, so it doesn't cause any problems. But here's the second side of this. We need it to bind to this major histocompatibility complex. But these major histocompatibility, blah, 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 these major histocompatibility complexes have a job. What a major histocompatibility complex does is basically take random proteins from inside the cell and express them on the surface. So here inside of this normal healthy cell are some normal healthy us proteins. Some of those normal healthy us proteins may be triangle shape, some of them may be circle shape, some of them may be square shape. These are all normal shaped proteins that this cell makes. And so what this cell is going to do is express a protein of ours, basically what you could call an us protein on its surface. So let's say it's taking the triangle one and exposing that triangle one on the surface, blah, blah, blah. Now, would we want to have a T cell in our body that was running around that could react and respond and build up a defense to proteins we own? No, absolutely not. We do not want that. And so the second half of this process is what we call the negative selection. With the negative selection, we need to make sure that the T cell does not respond to any of our antigens. So that first immature T cell we looked at, look at its binding site. It has the ability to bind to our major histocompatibility complex, but notice it binds to triangle shape antigens. So this particular immature lymphocyte, would we want to mature this and let this run around inside of our body?
No.
No.
Would that be an autoimmune disease?
Exactly. If this were to get out, that could cause an autoimmune disease where our body would be attacking itself. Absolutely. This would be an absolute fail. And so what would we want to do to that cell?
Destroy it.
Yeah, we'd want to kill it. However, notice if our cells, and again, obviously this is an extreme oversimplification. If all of our cells in our body only make 3 shapes of proteins, triangles, circles, and squares, if I have a lymphocyte that has the correct way to bind to our major histocompatibility complex, but then it has this wiggly binding site, is that going to bind to any of the shapes that we have?
No.
No.
No. So in that case, if it's not going to bind to any of our proteins, is it okay to mature this one and to let it become a T cell?
Yes.
Yeah. So this one passes. And when it passes, it matures into a T cell.
Does the fact that the thymus shrinks as we age have any impact on, like, does it produce less cells or?
It does produce less cells, but remember, your white blood cells are longer lasting. So you may be less adaptive to new pathogens that you're exposed to, but you've also built up a larger defense. So you have a larger reserve defense that way as well. So you may not be able to respond as well to something novel, which is something we saw with coronavirus. Who was impacted most dramatically by coronavirus when it first became prevalent? The more elderly people, or people who are immune compromised, because they weren't able to build up a defense as successfully as other people are able to do. So as our thymus shrinks, we do lose some of that ability to be able to respond and adapt. All right. These pictures are really simple. I appreciate that this is simplicity here, but I'm hoping these simple examples help it to make sense. So let's go back to the lecture and do the exact same thing. As I mentioned, there are two steps to the maturation of our T cell. The first is that positive selection, where the T cell must be able to bind to the cell. And the way that it binds to that cell is, again, by being able to recognize and bind to that specific self-protein, the major histocompatibility complex. But at the same way, we do not want it to recognize any of our self-antigens. So we do not want it to bind to any of the proteins or any of the antigens our cells make. Because as you guys pointed out, if this occurs, this is that autoimmune disorder where our body attacks itself. This ability to not react to any of our cells is a process we call becoming self-tolerant. Now, of all the cells that enter into the thymus, how many do you think are able to actually pass through this? Is this a kind of class where we want to give a generous curve, where we want to have lots of extra credit, where we want to get as many through and passing as possible?
Yes.
Really? Because it's okay if something maybe will respond to a cell in our body, so we just want to get it out there. Numbers are more important than having a cell that may respond to our body.
I just heard extra credit and was all for it. So no.
No, I appreciate that. In some things, it's a thing, but no, we want to have set a very high standard. Remember, T cells are the cells that attack and destroy other cells. That's not the kind of thing we want randomly running around the body willy-nilly. So we set a huge high standard to this. And of all the lymphocytes that come to the thymus for maturation, only about 2% pass. Now, the good news is this class has a much higher passing rate than 2%. But when we're talking about cells that are capable of killing other cells in our body, we absolutely positively want to set a high standard. So only about 2% of the T cells actually become immunocompetent. Only 2% mature. And of course, as we've talked about, once they mature, they will migrate back to the bone marrow, to the lymph nodes, to the spleen, right, to the tonsils, so on and so forth, liver, all of those things. So they're not going to stay in that thymus once they're matured. They're going to spread throughout the body. All right. Questions about becoming immunocompetent? All right, excellent. Oh, here's the pretty picture. So again, some go to the bone marrow, some go to the thymus to mature, become immunocompetent, and then they circulate throughout the body. But lymphocytes are not the only cells that are part of our immune response. Phagocytes are as well. And which are the two primary cells that are going to be our phagocytes in this immune response?
Monocytes and neutrophils.
Yep. Excellent. It's going to be those monocytes and those neutrophils. Remember, when we talked about our process of phagocytosis, remember we talked about how after it breaks down that pathogen, most of the materials from the digested pathogen are just recycled. But remember, some of them are used to stimulate an immune response. And we do this by presenting those antigens on the surface of the cell. What do you think we call cells that present these abnormal antigens on the surface of their cells?
Antigen presenting cells.
Antigen presenting cells. And because we can connect dots, what protein do you think that these antigen presenting cells use to express these foreign antigens on their surface?
The MHC.
Exactly. They use those major histocompatibility complexes to express these on the surface. Absolutely. Now, as we mentioned, indeed, our phagocytes are antigen presenting cells, but they're not the only ones. Dendritic cells, where were those dendritic cells found again?
In the skin.
Yeah. And even more specifically in the epidermis, but I would accept skin, absolutely. The dendritic cells, infected cells, right? Remember, an infected cell is a cell that is making proteins for a virus. So when it makes those proteins for a virus, and if it randomly is grabbing proteins from the inside and exposing them on the surface, it could be exposing those viral proteins on their surface. Our Kupffer cells are a special type of phagocyte that is found in the liver. We'll talk about those more when we get to the digestive system. But again, if you think about it, if we think of it in terms of where you're posting your guards, as we just finished talking about, in fact, some of you had as an essay question on the last exam, right? As we talked about, the arteries to the superior mesenteric and superior and inferior mesenteric arteries go to the small and large intestine directly. But remember, the blood from those don't go directly back to the inferior vena cava. They go to the liver first, where we can process all of that material that's being absorbed by the digestive system. Take it from being nutrient rich to being nutrient appropriate. Well, what else has the potential to get in while we're digesting that cheeseburger you had for breakfast? Harmful pathogens. So this can help us to protect us from any harmful pathogens that get in through our digestive system. So that makes sense to have those guards posted there in the liver. As we'll learn about, B cells also are antigen presenting cells in a special way. We'll talk about that a little bit later. But the one other thing, oh, I thought I had it here, but apparently I don't. Also, cancerous cells. One of the key things about a cancerous cell is that cancerous cell, not only is it dividing inappropriately and uncontrollably, but often cancerous cells make abnormal proteins. And if they're making abnormal proteins, then when those abnormal proteins are expressed on the surface, we can recognize something is wrong. Notice it's good that we're able to recognize all of these things. But there's one more thing that can be an antigen presenting cell that it isn't necessarily a good thing. If you've been drinking vodka for breakfast, lunch, and dinner for the past 2 years because of these COVID protocols, chances are you have damaged your liver and you need a new one. And when that new one gets put in, those new liver cells are presenting all of their normal proteins on their surface. However, are their normal proteins necessarily your normal proteins?
No, won't our body try to attack it?
Exactly, and that's the problem. If it's not compatible enough, if we recognize their proteins as being abnormal, then absolutely our body's defenses will attack that liver. That's why you can't just pick a liver off the street and stick it in your body. right? Getting a liver transplant, getting a heart transplant, getting a lung transplant are a very challenging process because some of these cells have as many as 200 different antigens that they can express on their surface. And luckily, not all 200 will cause a vigorous response. Remember, like with blood, blood has about 30, but only 2 cause a vigorous response. But if it's 20, how many people in the world have those exact same 20 proteins on their surface that you have on the surface of your liver?
Very few or
Yeah.
none.
Probably not that many, right? Absolutely, probably not that many. Notice it's more likely, because again, those things are determined how? Genetically. So you're much more likely to get a transplant from someone you're related to than someone random outside on the street. But even being, you know, even your mom, your dad, your sister, your brother aren't necessarily going to be compatible for a liver transplant. So that is the other challenge to this. The good and the bad of it is the system works really, really well, whether we want it to or not.
Professor,
Alright.
is that why people who, or most people who get like an organ transplant, they have to constantly be under antibiotics, and so their immune response is going to be lessened? It's like, I think that they have trouble traveling as well to certain
Well,
places.
yeah, not just antibiotics, but typically they're on some type of immunosuppressor. They're basically on some type of medicine that suppresses their immune response so that their immune system doesn't attack that organ. But you're absolutely right. The problem with that is it's not selective. So if we're suppressing your immune response, yes, it's not attacking your liver, but that also means it's going to have a harder time attacking that flu virus.
Right.
So yeah, so yes, you want to be limited with your travel, and you want to be limited with other things as well, because yeah, unfortunately, we're not able to just say, don't pay attention to that one. We have to suppress the entire immune response.
Right. Thank you.
Yep. No, great point.
I have a
Excellent.
question.
Yes, go ahead.
If this is way off topic, I'm totally fine. We can talk about it later. But I've heard people talk about like a cytokine storm with COVID, like either triggered by getting COVID, or some people use it as a reason not to get the vaccine. And I think it's just like an overactive immune response. But I was just curious what that was.
So as we will talk about, cytokines are a class of local hormones that are basically produced by our immune cells to help to stimulate an immune response. I haven't heard of it used in the term of a cytokine storm, but my guess is that
I think it's when one thing like just triggers it, like
well,
out
so
of.
yeah, so look, is it trying to think of the right way to answer this. Again, I'm not familiar with the term, and it's not my area of expertise, so I may be misunderstanding this, but the whole point of a vaccine or a booster of a vaccine, which is the thing that we're dealing with right now, is to generate an immune response. And with a booster, when you've already been exposed to something once, then you should get a more vigorous response. As we'll see, the whole point of these memory cells, as we've hinted at, is the memory cells allow us to produce a faster, a larger, a longer lasting, a more aggressive response when we're exposed to that pathogen a second time. So yeah, some people who are getting that third booster are spiking high fevers for a day or two, or feeling super achy and things like that, because it's stimulating an immune response to build up the memory cells. Because remember, as we talked about, you have to do the work to get the memory cells. But are you going to die from that 102-degree fever that you get as a result of that, or the achiness that you feel for a day or two? No, because you don't actually have the virus inside of you. In the case of a vaccine, you are getting it. So I don't, while you would be expected to get a vigorous response, like I said, I'm not familiar with the term storm, and so I don't know, and maybe there is some negative connotation to it that I'm not aware of, but that's kind of what you want.
Right, that's what I was thinking.
So
I think it's
again,
kind of
so
one
yeah,
of those
like I said, not my area of expertise, so there may be a subtlety to it or a potential risk associated with it that I'm not aware of, but I'm not aware of it.
Thank you.
Yep, no, it's a great question. Again, this portion of this class is something that is very poignant to all of us right now because we're all dealing with the repercussions of these types of things and the implications of vaccines and viruses and mutations and all the things that go along with it. So we're all living this right now. So I certainly appreciate and understand that. All righty. So as I mentioned, actually, this is a good stopping point. We've set the stage. We have these two types of immunity. We know how to stimulate the cells. We know what these two are going to be involved in. So now is a good chance to actually talk more specifics about all of these. But let's go ahead and take our first break. It's 1:06, so we'll take a 15-minute break. So we will restart at 1:21. And at 1:21, I will start the recording at that point. So any other questions? Those were all great questions. I don't claim to have all the answers, but I'm happy to attempt to answer them for you. Any other questions before we take our first break? All right, I'll see you guys in 15 minutes then. So our cell-mediated immunity, as I mentioned, is uses T cells. And this is where we get that hot cell on cell action. These are cells attacking other cells. Here we have the pretty picture from your textbook that does a nice job of showing how we have that immature lymphocyte goes to the thymus, and there it matures into T cells. And as it turns out, there are four main types of T cells that are going to be important for us. There are going to be helper T cells, and guess what helper T cells do? They help. There you go. See how easy that is? We have cytotoxic T cells that are also sometimes affectionately referred to as killer T cells. Guess what the killer T cells do?
Kill.
Kill. There you go. See how easy that is? Guess what memory T cells are used for?
Having memories, so like they're prepared for the future.
Exactly. And then regulatory T cells, which are also sometimes referred to as suppressor T cells. What do you think a regulatory or suppressor T cell does?
Does it keep them from over activating or?
Yeah, it helps to regulate the activity of the T cells or dampen them down, because you absolutely have the right idea. Once your infection is gone, do you want a bunch of hyped up cytotoxic T cells running around causing problems inside of your body? This is the part where you enthusiastically say no.
No.
No, exactly. No. So we need to stop the process once the process is over. So there you go. That's very convenient. They have the names that help us to understand exactly what it is that they are going to do for us. As I mentioned, we're talking about our T cells and our cell-mediated immunity because they typically are activated before the B cells. But one of the catches with these is they are not activated directly by the pathogen themselves. So they're not recognizing that harmful virus or that harmful bacteria. Instead, then, what is it that's going to stimulate our T cells? Those antigen presenting cells. And what are antigen presenting cells again? What's a common antigen presenting cell?
Phagocyte?
Yeah, phagocytes are a good example. But as we also talked about, pretty much all cells are able to be antigen presenting cells. Now, again, for our normal healthy cells, we don't care about those that are presenting, but the ones that are abnormal, the ones that are foreign, the ones that are, transplanted, things along those lines, those are the ones that are there, and those phagocytes. These are the primary sources where we're going to be getting those antigens presented to us.
What about monocytes and neutrophils?
That would be the phagocytes.
Okay.
And if you think back to what we saw in the T cell, there is this double recognition that is required. Our T cell needs to be able to recognize two things. They need to recognize the us protein, and that us protein is that major histocompatibility complex that we talked about. This is a protein that our cells have and express, but remember, they also have to recognize that abnormal, that foreign, that not us thing, and that is that not us antigen. So that antigen, from that virus or pathogen. And then again, we'll talk about abnormal and foreign and all of that, but let's just for simplicity talk about that pathogen antigen. So these are the two things that are necessary to be able to get our cell-mediated immunity started. We have to recognize that major histocompatibility complex and that pathogen's antigen. Now, we've mentioned this major histocompatibility complex a couple times now, but as it turns out, there are two main types of major histocompatibility complexes. They are the class 1 and the class 2. Let's go ahead and go to our whiteboard again for this. The first type is a class 1 major histocompatibility complex. These are expressed on almost all the cells of the body. And remember, these do exactly the same thing that those ones in the T cells do. Their job is basically to grab some random protein from inside the cell and express it on the surface. So if you think about this, the key to this is that it expresses an internal protein. So let's start easy. This is a normal cell in our body. Whoops. And let's go back to that super simple example we were using before. The normal, healthy, simple cells of our body make triangles, they make circles, and they make squares. So what's going to happen is my class 1 major histocompatibility complex is going to be hanging around here inside of the cell. And it's randomly going to pick one of these up. Let's say it picks up the square. And then it is going to be expressed on the surface. And so it is expressing that square on the surface. But then, like many of the proteins on the surface of the cell, they're dynamic. It gets reabsorbed. And then a new one gets posted on the surface. And this one's got the triangle on it. Notice in all of these instances, we have two things. We have the class 1 major histocompatibility complex, and we have our own self protein. Now, because our T cells have passed the negative selection test, are any of these normal proteins we're putting on the surface, are they going to stimulate an immune response? No. No. So the fact that they're on every single cell of our body is not a big deal because we've protected ourselves with that negative selection. However, let's change things up. Now, instead of a normal cell, this is now an infected cell. And now that this is an infected cell, it's not making normal proteins anymore. Now it's making proteins for our virus. And as we saw, those are squiggly topped. So now our type class 1 major histocompatibility complex is going to grab a squiggly top antigen and express it on the surface. And so now we have our class 1 major histocompatibility complex that is now expressing that not us antigen. And so now the right T cell is going to be able to come in. Can't be any old T cell. It has to be the T cell. Let's cheat a little bit, move this a little bit closer. That has just the right binding site where it's able to bind to our major histocompatibility complex, but it also is able to bind to our antigen. Notice it has, like we said, that double recognition. Where it is recognizing the foreign pathogen and the class 1 major histocompatibility complex. Questions on this? All right. But there is a second class of major histocompatibility complexes, and these are the class 2. These are the ones that are expressed on phagocytes. And these are also the ones that are expressed on B cells. Like I said, we'll talk about B cells and explain B cells when we get to them. As we know, what a phagocyte does is it grabs a virus or a pathogen from the outside, from the interstitial fluid. We know it destroys it. But remember, we then said how it takes some of its proteins and it expresses them on their surface. Now, notice this is a little bit different. These proteins that it are expressing, are these proteins that the phagocyte made?
Yes.
No.
Well, let's see. It found a virus that had abnormal proteins on it. It brought it in, it gobbled them up, and then it ripped off the arms and legs and expresses that arm and leg on the surface. Notice it didn't make it. It came from, in this case, the protein came from the pathogen. So notice what's happening here is this expresses an external protein. Notice over here, this is a protein over here with the class 1, this internal protein is a protein this cell made. Whereas over here, this is a protein the cell found. It found that pathogen, it gobbled up that pathogen, and it expressed it on its surface. Let's draw that phagocyte. All right, there was some pathogen floating around, and this pathogen just happened to have a squiggly shaped protein on its surface. This got endocytosed into the phagocyte. So our phagocyte brought it in, brought it into a vesicle. With the phagocyte inside of it. It then took its lysosome. With all of its digestive enzymes. Fuse the two of them together. And remind me again, when these things come together, what does it produce again?
Phagolysosome.
There we go. Makes a phagolysosome. Excellent. And during that process, my class 2 major histocompatibility complex is going to grab onto one of those abnormal antigens. And then when it does that, it'll express it on the surface. Notice again, we have a self part. That self part is the class 2 major histocompatibility complex. And the part 2, the not us antigen. That pathogen's antigen that it's now expressing on the surface. And so once again, a T cell is going to come in. That T cell is going to have the correct binding site to be able to bind to the class 2 major histocompatibility complex because it passed the positive selection. And it's going to have the right shape binding site to that abnormal pathogen. And so again, we get that double recognition. All righty. With me so far?
Yes.
Okay, if you're not, please let me know now because it is only going to get worse.
Actually, I have a question, Professor.
Yes.
What's the difference between the phagocyte and the phagosome?
The phagosome is the vesicle that contains the pathogen inside of it. So the phagosome is the vesicle inside of the phagocyte.
So the phagocyte will phagocytose the lysosome and then they will combine?
Remember, the lysosome is already a organelle that is inside of it. So it brings in the pathogen into a phagosome, into a vesicle. It already has the lysosome as an organelle floating around. So the lysosome binds with the phagosome and releases its contents into that phagosome to destroy the, right? So it's going to bind to it, release those digestive enzymes, and those digestive enzymes destroy the pathogen.
Which is in the phagosome.
Which is in the phagosome. Although once the vesicle of the lysosome binds to it, then we call it a phagolysosome.
Right. Okay. Thank
Yep.
you.
Yep. All righty. Any other questions? Like I said, it just gets worse from here. So we have to make sure we understand this.
So question, so in the infected cell, the MHC or the class 2 can also be present, that process.
No. Class 2 major histocompatibility complexes are only on the phagocytes. And B cells. But we'll worry about the B cells later.
So they
All
get
right.
the, sorry.
Go, I'm sorry, go ahead.
They get the proteins from whatever they like endocytose.
For the class 2, yes,
Yeah.
exactly. For the class 2, remember basically what that phagocyte is. That phagocyte is like any good outdoor cat. That outdoor cat's going to run around outside. He's going to kill a bird, and then he's going to bring it and lay it on your front porch so that you can see. It says, here, mommy, look what I found for you, right? Well, that's what my phagocyte here is doing. My phagocyte is saying, look, I just found this harmful pathogen. Go kill it, right? This is something I just found. So what this cell is going to say is this cell is going to say, go kill anything that has this protein. All right, don't kill me, but go kill anything that has this protein. But what about this cell over here, this infected cell that's expressing this abnormal protein on its surface? What is it saying by expressing this internal protein?
That I'm infected?
Exactly. It's saying, I'm infected. And if I'm infected, what should you do?
Kill
Kill
me.
me.
Kill me. So notice these two different classes of major histocompatibility complexes are giving two different types of signals because they're using two different types of proteins. The class 1 is saying, I'm making these abnormal proteins. There's something wrong with me, so you need to kill me. Whereas the phagocyte is saying, hey, I found this laying around. This doesn't belong. Go find anything that looks like this and kill it. Notice these are two incredibly powerful statements. Go kill those things or come kill me. Is that the kind of thing we want to do by mistake?
Definitely not.
Definitely not. Back in ancient times, cars had these things called keys. Well, first they had drivers, and then drivers had these things called keys. And the key wasn't some fob you had to have in your pocket that when you got 100 feet from your car, the car turned on, the door opened, the wheels rolled down. I mean, the windows rolled down, the seat warmer turned on, and everything else that happens. Back in ancient times, you had to actually put the key into the ignition. And as long as the key had the right shape and the ignition had the right shape, they would match up. But just putting the key into the ignition, would the car suddenly start?
No, you
No.
gotta turn it.
No. What else did you have to do?
Turn it.
You had to turn it. Exactly. So notice the double recognition is a starting point, but the same way just putting the key in doesn't start the car, the double recognition doesn't start the T cell. So for both of these types of T cells, we are going to need what, oh, no, hold on, I want to leave that dark. For both of these, we are going to need what we're going to call a co-stimulation. A co-stimulation is like the turning of that key. that co-stimulation is typically either going to be a special protein, either a special plasma protein, or a chemical signal. From the infected cell. So that's way too big. So let's make this a little smaller. There we go. So here on our infected cell is either going to have some type of chemical signal, or either a chemical protein sticking off the surface or some type of chemical signal. And as long as it is released at the same time as the double recognition, then our T cell is activated. All right. Notice if I just stand here with a car key in my hand and turn it in space, my car doesn't suddenly start. It only starts my car when I put it into the ignition and then I turn it. So both things have to happen at the same time. We need to have some type of chemical signal that is going to be a co-stimulation. All right. Questions on that?
Professor, so the class 1 would be third line of defense and the class 2 would be second line of defense?
This is all part of the third line of defense.
Because we talked about this phagolysosome and all that in the previous.
So the phagocyte gobbling up the pathogen, that is part of the second line of defense. Because when it grabs that abnormal thing and gobbles it up, it doesn't care what it is. However, when it takes its specific protein and puts it on the surface, and that stimulates a T cell, then that stimulation of the T cell is our third line of defense. Right? Remember, when it gobbled it up, it didn't care what its proteins looked like. It just gobbled it up. The T cell cares what the protein looks like.
So for some disorders, there is no first or second line. For example, for autoimmune, it's just third line of defense.
Well, you've got the right idea. With an autoimmune disorder, what happens, it would be kind of over here on the left. We have a T cell that is recognizing a protein that we make as being foreign, and it attacks and kills that cell. So yes, that's not, but again, that makes sense that the first and second line of defense shouldn't work with that because that's an abnormal function. It's not supposed to do that.
Okay, thank you. I was just trying to make connection with the autoimmune disease and.
Yep. Well, let's finish seeing how this works and then we can talk about when it goes wrong. All right. Any other questions? So notice there's basically three things that have to be together. We have to recognize the major histocompatibility complex and some foreign antigen. So that's our double recognition. But at the same time, we have to have that co-stimulation as well. Some kind of additional chemical signal from either the phagocyte or the infected cell. But notice one other thing. I know my drawing skills aren't the best, but basically these two T cells have the same binding site, squiggly on top. However, our two cells are giving two different signals. One is saying, kill me, and one is saying, go kill something else. So do we want the same T cells getting those same signals?
No.
No.
No. We want to have different T cells that do different things. And guess how T cells are different? T cells are different by having special types of proteins on their surface. These special types of proteins are called CD proteins. One example of a CD protein that all T cells pretty much have to have, we'll use pink for this one. Oops. Is a special protein that they have to have on their surface called CD3. If a T cell doesn't have CD3, it can't be activated. So if a T cell doesn't have CD3, it can't be activated, it's not going to be any good to us. So obviously all functional T cells have to have that CD3. But there are other CD proteins that can be different. For instance, one of these CD proteins, I'm running out of colors I haven't used, we'll go yellow. One of these CD proteins that hasn't been, we haven't identified yet, is a CD protein called CD8. So here we have our protein CD8. And in fact, this CD8 protein is so important for the function of this T cell that we actually call it a CD8 T cell. Other T cells may have a different protein, and that different protein could be, I'm going to use light blue. I'll just make it different shape so that we tell that these are different. A different protein on their surface, and that different protein on their surface is CD4. And that CD4 is so important that that's actually how we identify the T cells, CD4 T cells. And these two different types of T cells are going to have two different types of reactions. Our CD8, and I'm going to write this here, I'll cheat and do this. I make my cell a little smaller. Can you guys see if I write down here on the bottom? Can you guys see that?
Yes.
Okay. So our CD8 T cells primarily become the killer T cells, the cytotoxic T cells. Whereas, jiggle a little bit, sneak that up a little bit. Oops. CD4 T cells primarily become the helper T cells. And there's a bit of an easy way that I use to remember this fact. Notice over here, class 2 matches with our CD4. And of course, 2 times 4 is what?
8.
Whereas notice over here, our class 1 matches with the CD8, and 1 times 8
8.
There you go. Matchy, matchy.
Just to check, that's just for remembering these, right? There's no
That's,
significant.
yeah, that's just for remembering that the class 2 go with the CD4s and the class 1 go with the CD8. All right.
Okay.
I hope I've done an okay job of showing this on the board, but let's go through all the pretty pictures and all the pretty words from your textbook and make sure that this makes sense. So remember, as we talked about, our class 1 major histocompatibility complex is expressed on, again, let's not forget this, it's expressed on most cells. But like we talked about, while it's expressed on a healthy cell, do we care? No.
No.
It's only when it's on an infected cell that it's going to be presenting that abnormal internal antigen that we care and we want to destroy it. And again, your book does a nice job of showing the pretty process of how this occurs. Notice here it is making proteins for our virus. Some of those protein pieces are being picked up, added to a class 1 major histocompatibility complex and expressed on the surface. As I mentioned, you don't need to know this process. But there are two things you need to know. The two things you need to know is that we are expressing an internal antigen. And the other thing we need to know is what is the message that we're sending
Kill me.
with this?
Kill me.
Kill me. Right? So those are the two things we need to know about the class 1. The class 1 are expressing an internal antigen, and it's the cell way of saying there's something wrong with me, kill me. The class 2 are on phagocytes and B cells. We'll talk about those in a minute. But they express that external antigen. Again, here's a better version of that simple picture I was trying to draw. Notice again, here is our phagosome. Here's the lysosome binding with it, forming that phagolysosome, breaking it up into pieces. And once it's broken up into pieces, one of these pieces can be added to our class 2 major histocompatibility complex and expressed on its surface. Again, you do not need to understand the process. But there are two things you need to know. The first thing that you need to know is the class 2 expresses an external antigen. And what is the message we're sending here?
Kill others like this.
Yeah, go kill this. So like I said, your book does a great job of showing how these things are bound to the major histocompatibility complexes and expressed. I don't need you to memorize those processes, just understand the difference. Which cells express the class 1 and class 2, what type of proteins they express, and what type of message that is sending. Because as we've seen now, different T cells are going to respond to those different signals. Now, remember, as we mentioned, we have that double recognition. We can get that key into the ignition, but getting the key into the ignition isn't enough to start the car. Because we don't want to just randomly start up these T cells because they have the ability to kill other cells, and we don't want to do that randomly. So we need that third signal, a co-stimulation, right? It is separate from the major histocompatibility complex. It is from the antigen presenting cell. And it's either some type of plasma protein or some type of chemical signal that it is releasing at the same time. Okay. And again, starting a car is a serious process. You have to have the key in the ignition. You have to turn the key. Sometimes you have to have the brake pushed down before you can do it, right? The car's got to be in park. There's all these things that have to be in place before you can start the car. And it's the same thing here. There are all these things that have to be in place before we can start a T cell. Now, as I mentioned, there's not just one type of T cells. T cells, like all the cells of our body, have special proteins on their surface. And T cells, there are over 70 different CD markers that have been identified. Now, again, does that mean that every T cell has all 70?
No.
No, exactly. All T cells have different types. Some are more common. Some are less common. Some are more important. Some are less important. So again, there's going to be a variety of these on the cells. One of the most important ones is the CD3. As I mentioned, if a T cell doesn't have the CD3, it's not going to be able to be activated. So pretty much any functional T cell must have this one. But other T cells are identified by which of these two CD markers it has. Some T cells have the CD4, and as we already hinted at, most will then become helper T cells. Of course, what's the key word in that sentence?
Most.
Most, so that means not all. We'll talk about those other in a second. And most of the CD8 become the cytotoxic, or again, it is okay to call them killer T cells. Both terms are acceptable. All right, questions on that? All right, let's look at the pictures from your textbook then, similar to the ones I drew. Notice here, we have a CD8 T cell. So here is our T cell down here. Notice it has the CD8 markers, those CD8 proteins on it. It has its special binding site that allows it to bind to the major histocompatibility complex and to a specific antigen. And let's add some words to this page, right? This is a T cell, a CD8 T cell. So which class of major histocompatibility complex is it going to bind to?
1.
Excellent. What type of protein is this class 1 expressing? Is it an internal protein or is it an external protein?
Internal.
And what's the message?
Kill me.
Excellent. So notice we're getting our double recognition. We're binding to the class 1 major histocompatibility complex, and we're binding to the antigen. But remember that double recognition isn't enough by itself. it also requires from our class 1. Oh, that was the other thing. What cell is expressing this class 1 major histocompatibility complex?
An infected cell?
Yeah. It's an infected cell that's over here that is expressing this. And remember, it also has to express that whoops, no, I wanted a different color. That co-stimulation. So that with the co-stimulation and the double recognition together, this T cell will be activated. And when this T cell is activated, it is going to divide to make many copies. And most of the copies are going to become what?
CD8 T cells.
Well, they're all CD8 cells, but what do most of those CD8 cells, what is their job going to be?
Oh, killers. T cells.
Yeah, they're the cytotoxic. Most become cytotoxic T cells. Excellent. And again, I said most because a few of them are going to become memory cells and a few of them will become regulatory T cells. Notice we can do the exact same thing for the other type. We did that. CD4s, most become helper T cells, but some are going to become memory, some are going to become regulatory. And let's look at the pretty picture again. Here we have our T cell. In this case, its CD marker is the CD4 protein. This allows it to bind with the class 2 major histocompatibility complex. And again, we're getting our double recognition here. But let's do some work first. What type of cell expresses the class 2?
Phagocytes.
What type of protein is it expressing?
Internal.
Internal?
External.
External. And what type of message is it giving?
Find those like me.
Yeah, go find this, go kill this. This is around, go kill it. Don't kill me, go kill that. Excellent, so we have that. What's our picture missing here?
Co-stimulation.
Perfect, awesome, absolutely. We cannot forget that our phagocyte has to release that co-stimulation to also stimulate that T cell so that it can divide. And when it divides, as we talked about, most of the CD4s become helper T cells, whereas a few become memory and a few become regulatory. Excellent. Questions on that?
No.
So notice we've now seen the two main types of T cells, CD4 and CD8. And we've talked about how to stimulate both of them, how to activate both of them, all the things that go onto them, what type of message activates them. I mean, what type of protein activates them, what that message says, and all the things associated with that. So then what we have to do is we have to talk about what happens once a T cell is activated. However, I've thrown a lot of information at you, so this is probably a good point for another break to give you guys a chance to decompress and think about this. So let's go ahead and take one more break. It's 2:13 right now, so that means we'll come back at 2:28 and we will restart. And I will start the recording at that time. So any questions before we take our next break, our second break?
Yes, I had a question. I missed what was the co-stimulator for the CD4?
It's the co-stimulation is the same thing for both of them. So what's going to happen is our antigen presenting cell either has a protein that's bound to its surface that stimulates the T cell, or it releases some chemical signal. And that chemical signal spreads to the T cell and stimulates the T cell. So it can either be a membrane-bound protein or it can be a chemical. But in both cases, we call it the same thing. We call it a co-stimulation.
Okay, thank you.
Yep. Great question. Any others? All right, I will see you guys in 15 minutes. Any questions before we dive back in? All right, so we've talked about what needs to happen for our T cells to be activated. We need to get that double recognition, which includes, whoa, that's huge. Our major histocompatibility complex, a self part, and the foreign antigen, the non-self part. But on top of that, plus we also need some type of co-stimulation. And we've talked about all of that. So once all of that occurs, then as I hinted at, what's going to happen next is that we are going to get a very rapid division of that specific T cell. So that T cell that was activated, that has that very specific binding site, all right, our squiggly line binding site, is going to divide. And when it divides, it's going to produce many identical copies. Remember, as we said, this is antigen specific. So all of them are going to have the same binding site as the original. So they can respond only to the original antigen. So by getting those many copies, many daughter cells, and most of those forms are going to become active. There are a few that will not activate. They will stay inert, and they become our memory cells. And again, as we talked about, these memory cells will protect us, not from this infection, but from any future exposure to the same pathogen. So memory cells don't help us now, but their job is to help us in the future. Let's talk about the two different types of cells and what they do once activated. We'll start with our CD8s. Again, most of them come cytotoxic T cells. I think we've beaten the horse to death, so I don't have to make you guys say it again, but notice it is being activated by a class 1 major histocompatibility complex on the surface of an infected cell, getting that co-stimulation. So we have our double recognition and our co-stimulation, and it divides. A few of those cells stay memory cells, stay inactive, but the majority of them become our cytotoxic cells. And our cytotoxic cells' job, as we know, is to go and find and kill. It is going to look for any cell that is expressing that abnormal pathogen, and it will kill it. Now, it's going to do this in a similar way to what we saw with our natural killer cell. Again, it only will attack cells that are expressing the specific antigen on their class 1 major histocompatibility complex. So like I said, viral infected cells, cells that are infected with bacteria or pathocytes, cancer cells, transfused cells or transplant organs, those abnormal types of things. And like I said, they're going to work in a similar fashion to our natural killer cells. Remember what our natural killer cells did? They basically poked holes in the cell so that the cell lysed, or they triggered apoptosis. And we're going to see similar things with the cytotoxic T cells. One thing they can do is release a special protein. That protein is called perforin. And like we mentioned, this perforin is a protein that will line up on the surface of the cell. Oh, actually, that's horrible. Binds to the surface of the cell, and another one binds next to it, 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 and so on and so forth until they form a pore, they form a hole, allowing sodium in, allowing water in, causing the cell to fill and likely burst. It can also release a toxin like lymphotoxin, and lymphotoxin basically goes to the DNA of the cell and destroys the DNA. If a cell doesn't have DNA, can it make new proteins? Can it make new viruses? Can it repair itself or do any of those types of things?
No.
No. So it is going to destroy the cell by basically making the cell non-functional. And it can release a special protein called granzymes. And granzymes actually trigger the cell to undergo that programmed cell death that we talked about called apoptosis. All right. So like I said, similar to the natural killer cells. And remember, we said natural killer cells are a type of lymphocyte, a special type of lymphocyte. But this one can destroy the DNA. And of course, there are two bigger differences. This one is specific to the specific antigen. And unlike that lone little night security guard walking around by himself, remember the first thing this cell did is made 2,000 copies of itself. So instead of a single night watchman, you have a whole army of cytotoxic T cells running around killing anything that it can find that is expressing the specific antigen that activated it. And your book's got a really pretty picture that shows this. Again, not only are these going to indirectly, pardon me, not only are these going to directly kill the cell, but they can indirectly harm the cell as well by stimulating the cells to release a different type of interferon, what is known as a gamma interferon. Gamma interferons don't tell the nearby cells to protect themselves. Instead, they tell macrophages, hey, come here and gobble me up. So they can either kill the cell directly or they can actually indirectly cause their deaths as well. And here I think is the pretty picture that goes along with that. So notice our cytotoxic T cell finds that target cell expressing that specific antigen on that specific class 1 major histocompatibility complex. Exocytosis, the perforin, the granzymes, all of those, the lymphozymes, all those harmful things to destroy the target cell dies, and our cytotoxic T cell runs off to kill something else. Pretty straightforward. Questions on that?
Yeah, so just to be clear, the macrophages, it stimulates macrophages because it's big rather than other phagocytes because they eat smaller things.
Yes. Yeah, remember the monocytes are the macrophages, the big eaters. They're the ones that are large enough to actually engulf an entire cell.
Yeah, okay.
Yep. Great question. Any others? All right, like I said, I think that's pretty straightforward. It gets a little bit trickier with our CD4. We have to do a little bit more hand waving, but like I said, I know you'll get some of this stuff again in micros, so we can cheat with that a little bit. Notice again, CD4 cells, most are going to become the helper T cells. Notice in this case, our antigen presenting cell is instead going to be that phagocyte. Expressing that external antigen. It activates it with our double recognition. It activates it with, and again, I don't know why this picture doesn't show it, our co-stimulation. We can't forget about our co-stimulation. And just like the CD8s, the very first thing that is going to happen is it's going to divide. Most of the cells will stay active, will become active, but a few of the cells will remain inactive, becoming those memory cells that are going to help us in the future. But the big difference is what's going to happen now with this helper T cell. This helper T cell, once activated, is going to, actually, let's leave it here because after, I don't know if we talked about it. I looked up what you guys were talking about last time. We were talking about a cytokine storm. Our helper T cells are going to release a massive amount of chemical signals. And these are going to enhance and stimulate our immune response. All parts of it. So I was curious, so I actually did a search on a cytokine storm to see what exactly that was. And basically, it was kind of like what we had talked about. When your immune system is activated, these helper T cells release a massive amount of chemical signals that enhance and increase our immune response. If we're enhancing our immune response, then the more chemicals are released, the more it's stimulated, the more it's enhanced, which can lead to kind of a positive feedback process where even more are released and even bigger response. And while having an immune response is really, really good, as we've talked about, if you have too much histamine being released, for instance, that can cause something like anaphylaxis, where you get vasodilation, where you got a drop in blood pressure or a drop in heart rate as a result of it. Nauseousness can occur because of it. Fainting can occur because of it. So if these chemical storms become too significant, you can have too vigorous a response, and that can be the concern of that. Right. Luckily, especially, and again, when we're talking about it in terms of vaccines, usually, hopefully, it is a short-term reaction, but with an actual infection that can be longer lasting, it can be a little bit of a concern because the continued presence of that harmful pathogen can continue to stimulate even more and more and more. But making our immune response stronger and faster is definitely a good thing. And that is exactly what our helper T cells do. Like I said, cytokines are a class of, there's over 20 that have been identified. They're a class of local hormones. Normally we think of hormones that are released into the blood and they go everywhere. Cytokines are more local. So it is that B cell based, that helper T cell telling all the cells around it, hey, this is what's in the area. This is what we need to deal with. Help us deal with this issue right here. Interesting, one of the first things the helper T does is stimulate the helper T cells. Because if one helper T cell giving out chemical signals to coordinate our defense is good, then 10 is better. So it's going to stimulate helper T cells to divide and mature more rapidly. So we get more and we get them more quickly. Same thing with the cytotoxic T cells. Our cytotoxic T cells are going to mature more quickly. Get them on the line. Get those copies made and get them out there fighting, killing and destroying the cells that are damaged. But these chemical signals aren't limited to just our third line of defense. They will enhance inflammation. They will enhance the activity of the natural killer cells. They will attract phagocytes to the area. And if that's not enough, most, not all, but most B cells, as we're going to learn, they actually require these chemical signals from the helper Ts to be activated. So without helper T cells releasing their chemicals, we don't get our B cells activated. And if we don't get our B cells activated, remember the B cells are the ones that give us antibodies. So without helper T cells, we don't get antibodies. Makes these helper T cells sound pretty darn important, aren't they? The part where you say yes enthusiastically.
Yes.
Yeah.
Whew, good, excellent. So lucky for us, there is nothing that targets and directly destroys helper T cells. Because if something like that occurred, that would be really, really bad. But thankfully, nothing like that exists.
Wrong.
Wrong?
AIDS, HIV.
There we go. Excellent. I won't steal too much thunder from a group that is going to be presenting, but absolutely. One of our groups is going to be preventing HIV. And the human immunovirus, what does it target? CD4 T cells. It targets our helper T cells and can completely destroy a person's ability to defend themselves from their environment. Excellent. Here we did it with the words. Here we have the pretty picture. Notice here's our helper T cell binding to the class 2 major histocompatibility complex and the antigen, getting the co-stimulation, activating that helper T cell, which is then going to stimulate other helper T cells, stimulate the cytotoxic T cells, stimulate the B cells, and stimulate and enhance our non-specific defenses. So while a very simple looking illustration, it actually does a really excellent job, right? It shows our phagocyte engulfing a pathogen, taking the antigens, expressing it on the surface, and telling us everything that that helper T cell does. So for a very simple looking illustration, it actually does a great job of summarizing what we've been talking about. And so again, without those helper T cells, we are not able to mount a a vigorous immune response, which is what makes HIV so devastating. All right. Here we see the pretty picture summarizing this. Again, as we've talked about, and again, this is an older picture from an older version of your book. So notice, as we know, the CD8s are activated by the class 1 major histocompatibility complex. All right. Cell that is infected, releasing an internal protein. And again, most become cytotoxic T cells, some become memory, and some become suppressor. Notice this side of the picture shows how our class 2 activate the CD4 T cells. These are external proteins presented by phagocytes, and most become helper T cells, with a few becoming memory. But notice they don't have the suppressor here. Originally, it was thought that only CD4s produce suppressor. Then for a few years, we thought it was only CD8s. We now believe that both play a role in producing suppressor T cells. So the company line right now is both CD4 and CD8 can make suppressor T cells, or what are also known as regulatory T cells. So let's talk about those. How about we start with the first question? Which cells become them seem like a pretty easy question to ask. So why don't we know?
Because maybe they come from multiple strains.
True. Well, it turns out they do come from multiple strains. That does make it harder to be able to identify them. But also, let's think about what we're talking about here. We're not talking about a heart where we can take a frog and cut open its chest if it's paralyzed and actually watch the heart function. Or we can, with humans, use imaging techniques to be able to see the function of them in a live person as they're going. We're talking about cells and the chemicals that these cells produce and the interactions of those chemicals with the cells around them. Is that something that's easy to study in a living organism, let alone a human? No. These are things, in general, our immune response is a very difficult and challenging thing to study. And so the cells that actually turn it off are even harder to study. A tremendous amount of time and effort and energy is going into studying this, but we have a very poor understanding of it. And you can see the advantages to it. We actually talked about one of them earlier. As we mentioned, you get that liver transplant because you've been drinking vodka for breakfast, lunch, and dinner for the past, 2 years. And so you get that new liver. And as we talked about, one of the things that happens is you are then on immunosuppressors. You're taking chemicals, medicines for the rest of your life that are suppressing your entire immune response. As we talked about, the good news is that it lets you keep your liver. The bad news is it helps you fight something even as simple as the common cold. But what if we could target just the cells that would attack your liver? What if we could only suppress those cells so the rest of your immune response worked perfectly and we could just stop it from just targeting those one cells in your liver? That would be an incredibly powerful medication, right? So wanting to study this, wanting to understand how this works is hugely important. And like I said, massive amount of money and energy and time is going into this. Because if this is a nut we can crack, right, then really, if you think about it, we could then, it would revolutionize transplants. We wouldn't have to worry about someone matching for livers. We could randomly just pick some liver up off the street and stick it in your body. give you the right medicine so your body doesn't reject that liver, and everything else would be perfectly normal. All right, the abilities, the potential is tremendous, but it is incredibly hard and challenging to study. But let's talk about what we do know. These regulatory T cells are similar to helper T cells, which is why they originally thought they were just CD4s, because they release chemical signals. And in this case, the chemical signals are inhibitory cytokines. The normal cytokines enhance the immune response, so these slow down or stop the immune response. Because like we said, we've got all these hyped up T cells to help us to kill all of those infected cells. But once the infected cells are gone, we don't want a bunch of angry killer T cells running around bored looking for something to do. In fact, for a while, that was one of the main, I don't know if philosophy is the right word, but one of the beliefs was by many immunologists is that the dramatic increase in allergies that we've seen over the past 50 years are due to the increase in hygiene, right? Because now that our environments are more sterile, we wash our, our pacifiers after they drop on the floor, rather than picking up and shoving them back in the kid's mouth. We wash them out first. We wash their environments. We sterilize everything, and along those lines, that they thought maybe the immune system was getting bored. And because it was essentially bored, it was looking for things to fight. And since the only things to fight were around were things in the body, then maybe that was one of the reasons why there was an increase in autoimmune disorders. Maybe it's one of the reasons that there's an increase in allergies. I don't believe that philosophy is still held, but it was something that we looked at, right? So we don't want these activated immune cells being able to just randomly run around our body. Now, of course, it makes sense that we don't want to activate these right away. We want to activate these with a delay, because we want to give those T cells a chance to do their job and destroy the harmful pathogen. So they're going to be activated much more slowly than our helper and cytotoxic T cells. But eventually, we need to shut down the activity of our immune response and turn it off. And like I said, a better understanding of this could be an incredibly powerful tool in our future. All right, questions on that? All righty, so here's our summary chart, putting this all together, talking about the two different types of T cells, their activation, what they become, and all the fun things going along with that. All right, so any questions on our cell-mediated immunity before we go on to our humoral immunity? All right, as we mentioned, our humoral immunity is going to use the B cells. And those B cells are going to be used to make antibodies. Of course, antibodies is a common term. It is an acceptable term. I will allow you to use the term antibodies on the exam, but you should be aware of another fancy name for antibodies, and that is immunoglobulins. Immunoglobulins, as we talked about, are plasma proteins. These are proteins floating around inside of the blood and the other humors of the body, which is why this is called the humoral immunity. And lo and behold, like we've been drawing them, they are Y-shaped. However, notice they are a little bit more sophisticated than the Y shape we've been drawing. Notice there are two, hold on, leave that there. Change that to that. Ugh, stop it. There are two heavy chains. And two, oh, that's not how you spell heavy. And two light chains. But notice also there are two distinct regions as well. There is the part that I've drawn already that is known as the constant region. And this constant region is identical on all antibodies. However, notice at the end of the Y, kind of like we've been drawing since the very beginning of this, there is a variable region. And this variable region is also known as the antigen binding site. And guess what the antigen binding site does?
Binds antigens.
Binds antigens, exactly. Notice each antibody has two. And let's beat the dead horse. What determines the shape?
Genetics.
Genetics, exactly. So as we've been drawing, right? If you think about from when we were talking about blood, we drew this Y-shaped protein that had some different shaped binding sites at the end. And that's what we've been drawing. We've been drawing the constant region and the variable region. That variable region is where it binds the antigens, and the shapes of these binding sites is determined by our genes. So we have that constant region. We've got those four chains, the two heavy and the two light. And remember, the constant region, as the name indicates, is constant, is consistent, is identical for all antibodies. Which again, if you think about it, makes sense, because when we've been talking about these Y-shaped things binding to stuff, no matter what their binding site is, we've kind of talked about that tail as a flag that's being waved. If that tail is the same on every single antibody on your body, then notice your phagocyte doesn't care what the individual antigen is because it's not binding to the antigen. What is your phagocyte binding to, grabbing onto so that it can gobble it up? It's grabbing onto the tail. And all of those are the same. So it makes it very, very useful that way. And again, those unique binding sites are determined by your genes. Now, we've seen that antibody, that Y-shaped immunoglobulin, but there are actually 5 main types or classes of immunoglobulins, and they're identified by letters. The first, Ig for immunoglobulin, IgM, is the first one that typically responds at the site of an injury. These are the ones, remember, that we talked about that our C1 complement protein can bind to. Immunoglobulin M's are also the ones that cause agglutination. Someone remind me what agglutination was again?
Is that when your body reacts with the antigens of a different blood type?
Yep, exactly. Remember, we talked about how red blood cells will clump together, right? In fact, that's how we tested the blood types. We added the antibody, the immunoglobulin M antibody to it, mixed it up, and looked to see if the cells clump together or not. So absolutely, agglutination is that clumping together of the red blood cells, really of all cells, period. But usually we see it in the red blood cells. The second type of antibody is immunoglobulin A, IgA. These are the ones that are found in our glandular secretions. They're found in tears, they're found in saliva, they're found in the vaginal secretions, right? These are the ones whose job it is to help to keep pathogens out. If they can bind to a pathogen before, it can immobilize it, make it impossible for that pathogen to get its way inside. So it's part of that first line of defense that we talked about to keep things out. Immunoglobulin Ds, IgDs, are actually found on the surface of our B cells. We still haven't fully talked about our B cells and what they do yet, but we know those B cells need to be able to bind to and recognize foreign antigens. And it's these immunoglobulin Ds that help us to do that. Immunoglobulin G is by far the most common, making up about 80% of the immunoglobulins that are produced by your body. And these are the ones that are responsible for our primary and secondary humoral response. These are part of that passive immunity that mom can provide for a baby when she's in utero, which is why we talked about needing to take Rogan. If you're a Rh negative mom carrying an Rh positive baby, these immunoglobulin Gs can actually attack baby's blood, which would be a bad thing. And last, but definitely not least, are our immunoglobulin Es, our IgEs. These are the ones that primarily, instead of binding to pathogens, these are the ones that bind to our mast cells. These are the ones that bind to our basophils. These are the ones that trigger the release of histamine. Which is an awesome thing when you've been injured. However, as we've also talked about, if these are inappropriately activated, causing an inappropriate release of histamine, what do we call that condition?
Allergies.
Yeah, allergies, exactly. Or that hypersensitivities. So there are 5 main classes of immunoglobulins. And if you have trouble remembering that, just remember the mnemonic, good old Madge, right? Madge was, of course, my first love, right? Yes, I was 8, she was 52, but she was the waitress at the diner down the street, and she would give me shakes and the little metal cup along with the shake with the extra shake inside of it, and I loved her for that. So Madge will always hold an important spot in my heart. And she'll help me to remember my antibodies. All right. Questions on that?
Yeah, so the immunoglobulins basically stick onto things to flag them?
Great question. And that's exactly what we're going to talk about yet next. Now that we know the different types of antibodies, you are absolutely correct. We need to talk about what these antibodies do. Now, there is one more catch before we get to that. Again, every single one of these antibodies has that Y-shaped subunit that we've been talking about. However, for some of these antibodies, they are slightly different in how they're released. Notice IgEs, IgDs, and IgGs all are released as that Y-shaped subunit. So how many binding sites would we have with something like this? How many binding sites does my Y-shaped antibody have?
2.
Excellent. However, remember that IgA, that is the one that is released into the secretions of our body to stop that pathogen before it gets in. And notice that antibody is released as a dimer. And what's the advantage of having a dimer? How many binding sites does this molecule have?
4.
And when you're binding 4 things together, it clumps them together a little bit more. And again, it would make them harder to get in, or easier for them to get trapped in our tonsils, or easier for them to get trapped and swallowed down into our stomach, where the acids can destroy them, right? So having those multiple binding sites are even better. And then there's that IgM. It's the one that binds the complement protein and also does a really good job of clumping blood together. Why does it do such a really good job of binding and clumping that blood together? Because it's released as a pentamer. Not 1, not 2, but 5 antibody subunits all bound together. So how many binding sites does our immunoglobulin M have when it's released?
10.
And
So you can see with 10 binding sites how it would be so effective at being able to clump that blood together. Now, let's get to the question you asked. What is it that antibodies actually do? There are several things. Remember, the goal of an antibody is to, in general, target the pathogen. When it does that, it can do that and cause a couple of things. Some of the things that we've already talked about it being able to do, when it binds to the pathogen, it can cause complement proteins to come in and bind to it as well. And as we talked about, that can cause the cell to lyse. That can cause the cells to attract phagocytes, destroying the cell, destroying the pathogen. We talked about how it can clump blood cells together. If it clumps blood cells together, it makes it a bigger, larger mass, easier for our phagocytes to see and to be able to destroy. Now, agglutination is the binding together of cells. But remember, it's looking for the antigens. And some antigens can be water soluble. So if they're water soluble and we clump them together, they call them precipitation. Notice agglutination and precipitation mean the exact same thing. They're clumping things together. The only difference is whether or not it is clumping a cell together or it is clumping something water soluble together. But still, if it's clumping it together, it stops the spread and it makes it bigger and easier for things to find it so that our phagocytes are able to gobble it up. But I'm sorry.
I'm just confused why you'd want to clump the blood. Is it just like infected blood cells or like what's going on?
Well, remember, we don't want to clump our normal blood together, but if your blood type is O and I give you A blood, do you want that type A blood swimming around throughout your body?
No.
No, We want to clump it together and stop it from spreading and stop it from causing damage because we recognize it as being foreign and being abnormal, right? So our body's trying to do the right thing by protecting us, right? Now, the problem is if you're clumping a bunch of red blood cells together inside your blood vessels, is that necessarily a good thing to be having happening?
No.
No, right? Because it could block the blood vessels and cause all sorts of damage and blood flow changes and all sorts of other issues that way. But that's also why it's super important to make sure someone gets the right kind of blood. But notice there's one more thing here on this list our antibodies can do. In particular, with viruses. As we know, the way a virus works is a virus, and we'll make this virus a triangle here. This virus has to be able to bind to the plasma membrane of the cell to infect it. Well, what if our body starts making all these antibodies that bind to the surface of it and cover the surface of this virus? If I cover the surface of this virus with antibodies, is it going to be able to make contact with our cell and infect our cell?
No.
No. And so that stopping of the infection is what we call neutralization. And as we mentioned, these flags, these antibodies are then flags that attach our phagocytes to make it more likely to gobble it up. So notice in all of these ways, our antibodies help to stop the spread, stop the damage, and destroy those pathogens. Notice here with our antibodies, our goal is to kill the pathogen before it infects a cell. Once it affects a cell, then it's up to the T cells to deal with it. But antibodies' jobs are to stop the spread and to kill these pathogens before they infect the cell. All right. Questions on that? All right. Let's talk about how this process works. Let's talk about our humoral immune response. And you know what? Let's do this on our whiteboard. Notice we've talked about how to activate a CD8 cell, T cell, how to activate a CD4 T cell. And now we're going to talk about how to activate a B cell. Don't those three things sound like they would make really, really good essay questions?
Yes, definitely.
Yeah. Doesn't it seem highly likely that one of those three is almost certainly going to be an essay question on one of your exams?
Yep.
Yeah, absolutely. So let's talk about our B cell. Our B cell is a lymphocyte, as we know. It is a mature, immunocompetent T cell. Pardon me, B cell, a lymphocyte. Sorry, that's what I meant to say. Excellent. And we've already kind of hinted at some of the things that it might have on its surface. One of the things we know it's going to have on its surface are some major histocompatibility complexes. And does anybody remember which major histocompatibility complex we said would be on the surface of a B cell?
Class 1.
Was it 1?
2.
There we go. Remember, class 2 we said was on the phagocytes, but also on B cells. And again, in fairness, I kept telling you to ignore the B cell part of it. So it's okay if you didn't quite remember that. But now we remember it has and is expressing class 2 major histocompatibility complexes. And remind me again, which type of proteins do these express?
External.
Excellent. We also just learned that our B cells are going to express antibodies on their surface. Which antibodies?
IgD.
There we go. Ig, which one?
D as in dog.
There we go, IgD. On their surface. Each of our B cells is going to have a specific shape to their binding site. This particular B cell has triangles. A different B cell may have squares. A different B cell may have circles. And of course, what determines what shape these have on their surfaces with their IgDs?
Genetics.
Yeah, genetics, absolutely. So here we have all these different B cells with all these different shape binding sites, but here's the one we're going to be interested in. And as it turns out, what's going to happen first is our B cell is that it comes in contact with the antigen. So in this case, the harmful antigen that is floating around inside of our cell is the triangle-shaped one. And based on that, which of our 3 B cells that we have here is going to be able to bind to it? A, B, or C?
C.
I cheated.
Or B, B.
I made this the B cell because it's the B cell. So yeah, exactly. But you're right. It was the third one. Absolutely. So that antigen is going to bind to the immunoglobulin D antibody. Once that occurs, it actually endocytoses the antigen, and it is then going to express it in its class 2 major histocompatibility complex. So basically what's going to happen is it's going to bring this into the cell, and then it is going to express it on its major histocompatibility complex class 2. At this point, we say that the B cell is sensitized. Now, notice we didn't say activated. It's just sensitized. For most B cells, this is not enough to become active. There are a few that will become active, but for most B cells, need, well, let's actually change the way I wrote this. most B cells need help to become activated. What might be a good thing that could help a B cell become active?
Helper T cell.
A helper B cell. I mean, a helper T cell, exactly. And that's exactly what happens. A helper T cell, which remember we said are activated first. T cells are activated first. So we have a helper T cell. This helper T cell, as we know, is going to have a binding site that is going to allow it to bind to the major histocompatibility complex. And if this is an active helper T cell, then we know that it has the correct binding site for the correct antigen. And so what happens is, again, using that specific antigen, the already activated helper T binds to the B cell. When this occurs, our helper T cell is going to release chemical signals going to release those cytokines, and those cytokines are what activate the B cell. Questions on that?
Yeah, how does the cell pull the antigen off of the antibody like into the cell?
So what you have to remember is that cells' plasma membranes are dynamic. They're constantly expressing proteins and reabsorbing proteins. They're constantly changing their proteins on their surface. And you have to remember, the antibodies and the major histocompatibility complexes are not this big in relation to the cells. I'm drawing them this big because, again, it's convenient and we can see them. So these are tiny little proteins on the surface that are easy to be able to be endocytosed and expressed. So we're constantly breaking down and building up and changing our plasma membrane. All cells do that. In this case, it plays an important role in the activation of the cell. Okay. All right. Once activated, then not surprisingly, this cell is going to act in a very similar fashion to what we saw with our, whoops, hold on. I want that to stay red. Once the B cell is activated, what do you think that B cell is going to do?
Divide.
Yeah, exactly. It is going to divide rapidly and produce many daughter cells. These cells are going to be identical to the previous one. This is actually called clonal selection. It's the exact same thing that a T cell does. So why they call it clonal selection in B cells and not in T cells, I don't have a good answer for you because T cells produce clones as well. But you've got the right idea. It's going to divide and it's going to produce massive numbers of these T cells, all of them being identical to the one that was activated. So that means all of them are going to have that same triangle shaped antibody associated with them. Now, most of these cells will become active. But not all. Some of the cells will stay in an inactive state. And what do you think happens to those cells that stay in the inactive state?
They become the memory cells.
Exactly, they become the memory cells. Again, similar. So then the only question that remains is what happens to the rest of these cells. And I'll just draw this with one of it so that we can see. What happens with the rest of these cells is they grow quite large. These cells that grow quite large are called plasma cells. As we know, plasma cells have a nucleus inside of them. And aside from having a nucleus, the reason these cells grow so massive is because they just fill themselves up with this massive amount of interconnected, flattened membranous sacs with all these little dots all over the surface, which apparently I'm drawing as lines, but you get the idea. What, of course, am I drawing here?
Rough endoplasmic reticulum.
And in the interest of space, I'm just going to abbreviate it as rough endoplasmic reticulum. And what does rough endoplasmic reticulum do?
Protein synthesis.
Makes proteins. And that's exactly what this plasma cell does. This plasma cell produces and releases massive amounts of antibodies. So this starts making those IgG antibodies, those nice Y-shaped antibodies. And guess what binding site every single one of these is going to have? That triangle shape associated with the antigen that activated it originally. Let's go back to the pretty pictures from the textbook that show this. So notice here we have our B cell, a mature B cell, but a naive one that has binding sites determined genetically. In this case, that antigen binds to our IgD antibody. It is engulfed into the cell and then expresses that antigen on its class 2 major histocompatibility complex. At which point it is sensitized. However, as we mentioned, being sensitized isn't enough. Almost all of our B cells, there are a few that just being sensitized is enough to cause them to divide. But by far, the majority of them need the helper T cell to be activated. Notice here comes our helper T cell with the correct binding site for the major histocompatibility complex, with the right binding site for the antigen. And when it makes that binding, it releases those cytokines. And those cytokines activate the B cell. And that B cell undergoes clonal selection, which, like I said, is just a fancy way of saying it divides to produce a massive number of daughter cells. A few of those daughter cells stay inactive, stay memory cells, but the vast majority of them grow into these big, massive cells filled with rough endoplasmic reticulum that we call the daughter cells, that we call the plasma cells. And plasma cells make antibodies. Our IgG, which are the ones we said were responsible for the immune response, antibodies. And they're not messing around. A single plasma cell can make as many as 2,000 antibodies a second. Now, again, is that a number you need to memorize?
Oh,
No, of course not. I mean, it's useful if you want to woo women, right? Next time you're at a bar, walk up to a woman sitting there and there at the bar and say, hey, did you know your plasma cells can make 2,000 antibodies per second? And they just swoon when you say those things to you. And when you say those things to them. But other than that, it's not a useful piece of information. But the point is, massive amount of antibodies are being produced. Now, while it can keep that up for a time, do you think these plasma cells are going to be able to do that forever?
no.
No, they're going to exhaust themselves. So these plasma cells are relatively short-lived. At which case they pretty much just exhaust themselves and they die. So a plasma cell only lasts, once activated, a plasma cell only lasts for about 4 to 5 days. But let's think about that. 2,000 a second. How many seconds in a minute? How many minutes in an hour? How many hours in a day? How many in 5 days? How many antibodies is that?
Kind of a lot.
Exactly, a whole lot. I'm not going to bother doing the math, but I know it's a lot. This, what we just described, is what we call our primary humoral response. We are exposed to a harmful pathogen, some type of antigen, for the very first time. Our B cells get activated. Our B cells get sensitized. Then our B cells get activated. Then our B cells divide. Then those B cells become plasma cells. Then the plasma cells start producing antibodies. Is that going to happen instantaneously? No. So
No.
notice that's what our chart here shows, right? Before we've been exposed to our pathogen, we had almost no antibodies floating around in our blood. Notice they've just given us an artificial unit as that number, so they don't give us an actual number because, again, we're talking about crazy amounts. But notice it can take a week before we start to see significant amounts, and it doesn't peak till about day 10. Remember, one of the things we talked about with our third line of defense, our adaptive immunity, is it's relatively slow. So after you've been exposed to a flu virus, you're going to feel like crap for a few days, right? Because it's going to start to incubate. You're going to start to get symptoms. You'll have symptoms for a few days, but eventually, a little over a week and a half after you're exposed, you'll have produced a peak amount of antibodies. You're going to start feeling better. The amount of antibodies you're producing goes down, and about 4 weeks later, you're back down to about the original number of antibodies you had the last time. And as I mentioned, this is our primary humoral response. What happens the first time we are exposed? Excellent. Very useful process, but there's one thing that we're forgetting about. While many of the cells that are produced become plasma cells, some become memory cells. And remember, as we've talked about, those memory cells are going to help us if we're ever exposed to that pathogen again. So let's do that. Let's expose ourselves to that pathogen a second time. When we get exposed a second time, we get a secondary humoral response. And look at the difference here. Notice immediately right away within the first day, we're already producing more antibodies than we did the entire time that we were exposed to it the first time. Notice by the first week, we're almost producing 100 times more antibodies than we did for the first exposure. So notice how much faster and stronger this response is. And remember, by 4 weeks, our antibody levels were back to where they were before. Is the same thing true here?
No.
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 Not 4 weeks, not 5 weeks, not 6 weeks, not 10 weeks. So not only is it faster, not only is it stronger, but it is a much longer lasting response. And the advantage, the biggest advantage to this is so fast and so strong that in all likelihood, no symptoms or very minor symptoms are felt. So,
Professor?
yes.
Let's just say like there are 3 types of memory B cells in your body that correspond to 3 different like bacteria or like a infection.
Okay.
If you got infected with all 3 at the exact same time, would that affect the efficiency of any of the other B cells? Or can all different types be working at the exact same time at full efficiency?
So what I would say is probably the only limiting factor are the resources, your amino acids, right? There is no reason that all 3 of them can't all be working at the same time, because each one is activating a different memory cell, and that different memory cell is going to divide. And so all these processes, so there's no issue about that. My initial response was no, it wouldn't have any effect at all. However, when you're producing that many antibodies, obviously you have to have enough amino acids to be able to produce all of those. So I guess my only thought is that maybe it might be slightly less because of the limited of resources, but my guess is that it probably wouldn't be a significant drop.
Okay, got it.
So yeah.
Thank you.
Yep. Yeah, there's nothing that would stop the cells from dividing and doing everything normally. Each one works independently. So there's no issue there. I guess, like I said, so my initial response is no change in any of their effects, except from just maybe a resource standpoint. You might be limited on how many amino acids were available to make protein. So maybe there might be a small decrease because of that, but otherwise shouldn't have any effect. Again, to beat the dead horse of the difference between these primary and secondary, I've got a couple other pictures that show this as well. Again, notice the point of this one. At this point right here, we are exposed to a pathogen, right? So here's pathogen A. We get exposed to pathogen A, and then here at day 28, we get exposed to pathogen A second time, but we also get exposed to pathogen B. So notice, as you mentioned, the A cells that have memory cells, because they're those memory cells, are going to give us a much more exaggerated, fast, rapid, strong response to A, whereas B, it's our first exposure. So notice the path of B antibody production is the same as A was originally. So yeah, so the pathogens shouldn't interact with each other significantly. And then again, here's our pretty picture visualizing what's actually happening with the B cells. All sorts of random B cells, but notice it's only the B cell that is specific to the antigen that divides, making a bunch of plasma cells, but also making memory cells. so that if we're exposed to that same pathogen a second time, we get a much more vigorous response. Faster, stronger, longer. And that second response is typically so aggressive that you rarely see symptoms, if any at all. All right. Last thing I want to leave you with is this great picture from your textbook. That page number may be off because I think this was from I wrote that during the previous edition of the book. But notice the nice thing about it is they've shown all of our lines of defenses, our innate defenses, the surface barriers, the internal defenses, the adaptive defenses, how they relate to each other, how all of this stuff works together to provide us with that overall body defense. All right, questions on this? All right, I know, well, see, again, this is one of those things, here's all the tables. This is one of those lectures that nobody's really happy with. If you have taken micro, then you know I've just touched the surface of this information, right? And so you were probably bored by all the things that I was saying. If you haven't taken micro, this information can be really overwhelming, and it seems like a lot. So half the class is terrorized, half the class is bored out of their mind. So like I said, nobody likes this lecture, but it is important. We do need to work our way through it. And the good news is you have 5 days to decompress and make sense of this information. And I definitely want you to do that. However, the other important thing that you need to do to prepare for the next class is to start looking at the endocrine system and the study guide that I've provided for you. I'm sure all of you have printed it out already, and if not, first and foremost, shame on you. But let's take a quick look at it. There we go. Under the endocrine anatomy lab handouts, is this study guide. This study guide contains the information you are going to be responsible for on the next exam for much of the endocrine system. At the end of this handout is all of the histology that you're going to be responsible for. And notice in parentheses are the key terms that I will use to help you to identify that. You do not have to color this and turn this in. If you took me for 430, you know that I had you do that with the epithelial tissues and connective tissues, but I did that to emphasize to you how important histology is. Hopefully at this point in your anatomy and physiology career, you appreciate how important histology is. You'll also notice that not all of the glands are on here. Notice, for instance, the thymus isn't on here. Notice I didn't hold you responsible for the thymus histologically when we talked about the lymphatic system, and I'm not doing it here either. Clearly, when you talk about the ovaries and the testes, there's a whole lot more to the anatomy of those that we'll worry about and talk about when we get to the reproductive system. But this is enough information to get us going with what we need to talk about for this section. So there's your histology. You've got lots of resources to help you with that. But what I also want to talk about is this part of the handout. Notice this part of the handout has the glands that you're responsible for, the hormones you're responsible for, and then 3 additional pieces of information. What that hormone targets, how that hormone is regulated, and what the function of that hormone is. Let's talk about one that might be easy, that most of us might have some familiarity with. Insulin. Insulin is produced by the pancreas. Does anybody know what insulin actually targets?
Glucose?
What about the glucose? Does it actually target the glucose itself?
Glycogen.
Well, you get getting closer. It has to do with glycogen. It has to do with glucose. So when we think about the function, it plays a role in the regulation of glucose levels. So your little kid goes to that birthday party back in ancient times when we could have birthday parties. And while there, they had 2 pieces of cake. They had 3 scoops of ice cream. And as a going away present, they gave you a bag full of candy to take home as your party gift, right? So after ingesting all of that glucose, what does your pancreas do?
Releases insulin,
And what does that insulin tell your body to do?
break down the glucose.
Well, you're really breaking it down. What are you doing to the glucose?
Storing.
Cell.
You're storing it. Basically, it targets most of the cells of the body. And it tells those cells of the body to take the glucose out of the blood and store it. Right? Now, I do say it affects most of the cells of the body. Are there any cells in particular it might target more, have a bigger impact on?
Muscle cells.
Excellent. Skeletal muscle and anything else?
Maybe GI.
Close. It is a digestive organ. What's the other digestive organ that might be sensitive to insulin levels? There we go. Juan's got it. Liver. So notice we were able to identify the targets. We were able to identify what its function is, regulate glucose level, and now we can be more specific with that. It stores glucose in the cells. And what effect does that have on glucose levels in the blood?
Drops.
There we go. So it drops glucose levels in the blood. Excellent. What that leaves us with is regulated. Regulated is going to take a little bit of explanation. So we'll get a chance to talk about it, although we might be able to figure it out in insulin. What is the change in the body that causes us to produce insulin? What did that kid do that caused their body to release that insulin?
He ate the cake.
Ate the cake. So notice it's the glucose levels in the blood that triggered the release of the insulin. So notice it's regulated by glucose in the blood. I don't expect you to figure out all the regulations on your own. We will go over this table together. The entire table, we will go over it together. But I think it's important to start thinking about it and looking at it first. So if nothing else, start trying to figure out what the targets might be and what the functions might be of all these hormones. And then together we'll work on the rest of it. We'll work on the regulated part. Okay. So this is something that I encourage you to start looking at now before Tuesday's class, because Tuesday and Thursday, basically, we're going to be powering through this list. That is what we're going to be doing. It is boring and exhausting and overwhelming because it is a ton of amount of information that isn't always necessarily straightforward and isn't necessarily interesting as well. So it is easy to get overwhelmed by it. So taking time to prepare ahead of time will definitely make this easier. So while absolutely positively, do not forget about the lymphatic system. Absolutely positively use the next 5 days to focus on your body defenses that we talked about. But you also might want to use the next 5 days to be looking ahead as well. All right. I'm sorry?
I just had a quick question.
Yeah.
If we go to the endocrine system chapter in our book, will it give us all this information or is it kind of more spread out?
Yeah, no, it will definitely be, it should all definitely be in there. Absolutely. The one caveat that I would give for that is when you're doing histology, For instance, if you want to, I'm sure the endocrine system will have some of these things in there. But for instance, if you want good slides of the pancreas, you can look in the endocrine, but you might also want to look in the digestive system section of your histology atlas. Same thing for the gonads, right? If you want, I'm sure the endocrine probably has testes and ovary slides, but if you want even better pictures and better explanations, then you might want to consider going to the reproductive system in your histology atlas. So for the histology, you might want to look around in your atlas to a couple other organ systems that make more sense. But certainly your endocrine chapter in the textbook will talk about all these things. Yes.
Got it. And then the targets regulated by functions, that is for each one of the hormones, right?
Absolutely. Yeah.
Got it. Thank you.
Notice we just did all three of those things for insulin. Oh, there's one other thing I want to point out on this list. Not here, here. Excellent. Notice there's one other thing, subtle thing you may not notice right away when you're looking at it, but notice when you look at the hormones, androgens are a class of hormones. There are almost a dozen different androgens that are produced by the body. Am I holding you responsible for knowing all 12 specific androgens that are produced by the body?
No.
No, but am I holding you responsible for the most common specific type of androgen?
Yes.
And what is the most common androgen that is produced by the body?
Testosterone.
Excellent. What is the most common estrogen produced by the body?
Estradiol.
Yeah, exactly. If we go to, I got to go back upwards. For the adrenal gland, in the adrenal cortex, there is a class of hormones called glucocorticoids. What do you think the most common glucocorticoid is?
Cortisol.
Yeah. So notice for some of these hormones, you need to know both the classes of hormones produced and at least one specific example of a hormone. And usually the specific example is typically the most common.
And Professor, the information, like the targets regulated and the function, you want that for the entire class or the most specific one?
Both. When we
Okay.
get to the glucocorticoids, you'll tell me how it's regulated or what it targets specific, what its general function is, but you'll also tell me the specific function of cortisol.
Got it.
Okay. Now, again, some of those it's a little more straightforward than others. The other thing that I want to emphasize about this is, so on the exam, for instance, on the, we'll take the thyroid gland because the thyroid gland's easy. I could have a picture of the thyroid gland and I could have an arrow pointing to the thyroid gland here, one of the cells of the thyroid gland. And if you think about it, I can ask all of those questions. Identify the hormone that is produced by this gland. Identify the target of the hormone produced by this gland. Identify the function, the specific function of the hormone produced by this gland, and identify how hormone production in this gland is regulated. So notice all of this information on these handouts, the targets, the regulated by, and the functions. These make good questions on a lecture exam, but I guarantee these will also be things that I ask you on the lab exam as well. I can point at one of these cells and I could really ask all of these questions. I could ask you, what gland is this? I could ask you, identify the hormone that is being produced. Identify the target. Identify how it's regulated. Identify its function. So really, all 5 of these are things that I could ask on a lab exam by pointing at a gland.
But
All right.
Professor, if you were to like point at the thyroid gland and you'd say like what hormone, since there's two, you want us to put 6 different answers, right? And like the target's regulated function for both.
No.
Oh,
So let's cheat. To do this cheating, we are going to do a simple version of this. And I'll be honest, this is actually a picture I don't even like. But we're going here at the Casumas River College Virtual Anatomy Lab. We're going to find the endocrine system, where we're going to look at the endocrine chart. There we go. One of the things that you'll notice is, for the most part, these glands look pretty different from each other. So it's going to make it a little bit convenient to be able to tell them apart. This happens to be the thyroid gland. And if you'll notice, the thyroid gland basically has 2 types of cells. It has all these cells forming a follicle that contains this fluid. But if you notice also, there are these cells off to the side. I know our artist has kind of made them look like smooth muscle cells, but they're not. 2 different types of cells. So notice, if, for instance, I were to have an arrow that I were to be pointing at this purple cell and ask you to identify the hormone that is produced by this cell, but I was also to take this arrow and change it and point here instead and ask you to identify the hormone produced by this cell, do you think these 2 cells produce the same hormone, or do you think they produce different hormones?
different.
Yeah. So that's how you have different answers. Both of these 2 cells produce different hormones. Now, the good news is 1 gland, so we know it's the thyroid gland, but one cell is going to produce one type of hormone, the other cell is going to produce a different type of hormone. Each
Got it.
one has their own function, their own targets, and they're both regulated differently.
Thank you.
No problem. It's a great question. And like I said, we will go over that entire list together. I'm going to make sure you understand this material. But as you see, that list is long. And like I said, it isn't particularly fun or interesting. So like I said, it's going to be long, it's going to be exhausting, it's going to be boring. So if you spend any time looking at this first, it will definitely help for next week. All right. Excellent. Any other questions? All right, and that is all I have for you today. So the rest of your time is yours. Start your weekend early, study hard. Have a good weekend. Let's actually